Hello and welcome to 2 fatty acids. In today's video, we will discuss one category of macromolecules, one of high importance, and that is carbohydrates, carbs for short. Let's get to it. So first off, what is a carbohydrate? A carb is a molecule made of the atoms carbon, hydrogen, and oxygen, often in a 1:2:1 ratio, meaning that most carbs are multiples of the molecule CH2O. Carbs are joined to make up sugars, and they function in the body through providing quick energy, support, and structural protection, and nutrients for the body to build upon once broken up by metabolism. Carbs also play an important role in cell processes such as cellular respiration and photosynthesis, which will be touched upon in later videos. There are many different types of carbs. Monosaccharides are single carbs of a specific type, not conjoined with any other carbs. To help remember this, keep in mind that mono means one and saccharide means sugar, so monosaccharide means one sugar. Similar to how mono means one, di, means two. So disaccharide means two sugars and is used to describe two carbohydrates joined through a covalent bond. Don't forget, a covalent bond is when two molecules share an electron, which differs from ionic bonds, where one molecule gives away electrons for the other to accept. The difference is that while ionic bonds affect the charge of molecules, covalent bonds do not. It is important to note, however, that both bonds bring molecules together, although at different strengths. Technically, the specific covalent bond between two carbohydrates is referred to as a glycosidic linkage. This covalent bond, as well as other covalent bonds in different macromolecules, is formed through dehydration synthesis, which we will discuss a little bit later in this video. As poly means many, a polysaccharide means many sugars and refers to more than two sugars joined through bonds to form a larger sugar molecule. Before we move on to diagrams of carb structure, you will need to know the terms monomers and polymers. Monomers are the individual pieces that make up polymers or larger pieces. In this case, monosaccharides are the monomers and polysaccharides are the polymers, formed when more than two monosaccharides join together. Now, the different types of monosaccharides are classified by the number of carbon atoms in the molecule and the carbonyl group placement in its structure. You will need to know all the functional groups for the AP Biology exam, and the carbonyl group is one of these. As shown, it's a carbon double bonded to an oxygen. We will make a short video soon on the necessary functional groups to know. Carb monomers can be either linear or ring-shaped. Triose is an example of a linear sugar. The name triose comes from tri, meaning three, since this molecule has 3 carbons, and ose, which is often the ending for sugars. Hexose, aka glucose, is an example of a ring-shaped sugar. Hex comes from it having 6 carbons, and ose, again, comes from it being a sugar. It's good to pay attention to the endings of biological words, as this can help decipher their meanings. While sugars usually end in ose, enzymes usually end in ase. Of course, there are exceptions to this rule, so it's better to know the names of specific sugars or enzymes. This next diagram shows dehydration synthesis using an example of disaccharides. In dehydration synthesis, a water molecule, H2O, is removed from the space between both molecules, allowing these molecules to covalently bond to each other with the new space they have. This water molecule is formed by taking a hydrogen from each hydroxyl group, OH, on the ends of the molecules, and one additional oxygen from one of the hydroxyl groups. This forms H2O and leaves one oxygen left for both molecules to share. Continuing on with the structure of carbs, we will now talk about polysaccharides. Polysaccharides vary in structure based on the placement of their hydroxyl groups. There is alpha formation, represented by A, where the hydroxyl group is continuous in one direction throughout the polysaccharide, and the beta formation, represented by B, where the hydroxyl group goes back and forth between its orientation on the polysaccharide. Due to the different placement, alpha glucose is helical and beta glucose is linear. Additionally, as structure impacts function in every biological sense, certain enzymes can hydrolyze alpha linkages but not beta linkages. This is why cellulose in plants, which is made of beta linkages, is not digested by humans and instead becomes stool. Now that you have an overview of carbohydrates, here are some important carbs to remember. Glucose, also known as hexose, which we saw earlier, is a monosaccharide ring used for quick energy. It has a role in respiration and as a reactant and a role in photosynthesis as a product. Starch is a helical polysaccharide specific to plants, which stores energy. The simplest form of starch is called amylose. Glycogen is a helical polysaccharide specific to animals, which also stores energy. Cellulose is a linear polysaccharide, which forms the cell wall of plant cells, providing structural support for plants. Humans cannot digest cellulose, as previously mentioned, and this is due to the hydrogen bonds it forms with itself. The cell wall, which will be discussed more in depth later, also regulates the amount of water coming into the cell, which can help prevent cells from bursting. Finally, chitin is a polysaccharide in the structure of arthropods, which helps provide support. It will be helpful for you to keep these terms and definitions in mind, as they will certainly come up later in the course. Thank you for watching, and don't forget to hit subscribe, like, and comment for more biology content. See you next time, and stay hungry for knowledge.